Hello, everyone. My name is Thi. I'm from the Hugh Kaul Precision Medicines at the University of Alabama at Birmingham, and today it's my pleasure to present a Medikanren-based drug repurposing approach using the transcriptome data from patients with CHAMP1 disorder. Here is the outline of my talk. I'm going to give a little bit of background about drug repurposing for rare genetic disorder, and then RNA sequencing data as the emerging type of data for this purpose. I will provide a brief introduction into Medikanren, our AI tool, and the duet query used for finding drugs, knowing the target genes and the phenotypes. And lastly, I will present the result of this workflow on Jepson, the potential use of metformin for this disorder. So first of all, why drug repurposing? Traditional drug discovery typically find one drug that target one particular molecular target for a particular disease indication. However, we know that a disease, even caused by one single gene, may affect more than one gene target, and a drug, even though originally intended to modify one target, may have downstream effects on multiple genes and organ tissues. These polypharmacy effects of a drug can be harnessed for drug repurposing. Traditional drug discovery and development is a very complex process that can take between 10 to 17 years and cost more than $1 billion to bring a drug to market for a particular disease indication. Typically, drug development has two phases: the preclinical phase and the clinical phase. In the preclinical phase, basic research is conducted to find the target, and then drugs are developed to act on these targets. In this phase, we learn about the pharmacology and toxicity of a drug. In the clinical phase, the drug is tested in human. Phase 1 tests the safety dosage. Phase 2 and 3 determine both safety and efficacy. Most drugs actually fail at phase 2 and phase 3. Now, in rare or orphan diseases, there isn't enough of return of investment to do traditional drug discovery process. And so drug repurposing has emerged as a fast approach to identifying compounds with already known pharmacology and toxicity profile without having to go through the lengthy preclinical phase. There are about 9,000 drugs that known drugs that treat about 5,000 known diseases. However, there are 7,000 rare diseases, and many of them we don't have available therapies for due to little interest to make it happen. In terms of target, among the 25,000 human genes, 400 are known as drug targets. I believe if we can mine existing knowledge about drug, disease, and gene targets, perhaps we can help find a fast and economical way to for treatments of rare genetic disorders. In terms of I want to explain a little bit about the patient RNA sequencing data. The central dogma of molecular biology explains the flow of genetic information within a biological system. DNA code is transcribed to RNA, which is then translated to protein, which play major function in living system. More frequently, high throughput approach to diagnose rare genetic diseases are whole genome sequencing and whole exome sequencing. And whole genome sequencing look at the entire genome to identify rare variants that may cause diseases. Whole exome sequencing only look at the protein coding regions of the genome. RNA sequencing is another high throughput technology, and allows looking at the entire gene expression of the genome, essentially to see what genes are being turned on. Even though whole exome sequencing and whole genome sequencing are more common, and they are routine procedure to help diagnose rare genetic disorders. The current diagnostic rate is only 25 to 50%. RNAseq data emerge as a complementary tool to increase the diagnostic yield. However, there are many considerations to use patients' RNA sequencing data for diagnostic as well to find targets for drug repurposing. So first of all, what genes are turned on? Depending on the particular cell types, tissues, and environment, different cell types that make up different tissues have different structure and functions due to their different gene expression profile. A neuron has completely different gene expression profile than a skin cells or a bone cell, even though they all share the same DNA code. The same cell type also turn on different genes depending on the different stages of their development. So gene expression is very dynamic in time and space. And so the RNA-seq data only represent a snapshot of gene expression in a particular tissue or cell type at that time point. Therefore, it is important to sequence the disease-relevant tissue. And oftentimes, it is challenging to get access to disease-relevant tissues, particularly for neurodevelopmental disorders. So it's rare that we can get biopsies of a patient brain, for example. But alternatively, we can get transcription data from cell culture or organoids, which may reduce a lot of noise compared to profiling in situ. So in terms of the CHAMP1 genetic disorders background, I presume everyone in this conference is well familiar with this genetic disorders. But briefly, CHAMP1 stands for chromosome alignment maintaining phosphoprotein number 1. It encodes a zinc finger protein that regulates the proper chromosome segregation during the metaphase of the cell cycle. And this genetic disorder is typically caused by de novo mutations, would lead to loss of function of important domains that affects the protein that affect protein function and affect cell division and brain development. So the case I was working on is Jeffson case, the PMI 18-4 case. His son has a frame shift mutation, would lead to truncation of the protein and loss of several important domains of the CHAM1 protein, including the zinc finger domains. And we expect this lead to loss of function of the protein and a defect in cell division and neurodevelopment. There's also a list of his phenotypes. So the source of data for this project are culture fibroblasts. We were isolated from the skin biopsies of CHAMP1 patients or control. The skin biopsies were then digested and isolated to obtain a pure population of fibroblast cells, which could be expanded in cell culture condition in the lab and then stored in liquid nitrogen for future use. Here is the metadata for the transcriptome dataset provided by Dr. Lessel and Dr. Hakan. I analyzed these two datasets separately, and then I also compared the drug results in the end. Just a few words of caution about the type of data, as these are data derived from patients' fibroblasts. So these cells have been sitting on the plate in cell culture conditions. There are potential artifacts. We are mostly interested in the difference in gene expression that are caused by different CHAM1 variants to see the variant impact on the transcriptome. But this difference could be due to other differences that are noise that we're not interested in. It could be due to different age, sex, ethnicity of the samples. Also, it could be due to different cell culture condition, and also their RNA extraction, library preparation, sequencing platform. Next, I'm going to give a brief introduction into our AI tool, Medikanren. Medikanren was developed as part of a multi-institutional project funded by the NIH and CAT called the Biomedical Data Translator Program back in 2016. This translator program was created to address one of the biggest challenges in translational science. It aimed to create a unified system capable of integrating existing all biomedical knowledge base to accelerate translational research. It's a very ambitious project from the NIH. So as the number of data and articles are being deposited are growing, there's a tremendous challenge to be able to connect knowledge from these different knowledge sources and then to link them and use them to advance medical research. At the Hugh Precision Medicine Institute at UAB, our team is working on Medikanren, an AI tool that can assist biomedical researchers to be able to reason across different biomedical knowledge bases. You can think about Medikanren as a giant knowledge graph that contains knowledge from all existing databases about drugs, genes, diseases, molecular pathways, environmental exposure, and their relationship with each other. Medikanren also contain a system that allows querying these linked knowledge graphs. Medikanren is very much like a Google, but for structured biomedical knowledge. We routinely use Medikanren at PMI for clinical case reviews for rare disease patients. You can use Medikanren to ask complex questions such as what FDA-approved drugs decrease expression of gene A and or modify disease X condition. For the drug repurposing workflow, for this case, I use a 2H query to look for drugs that can reverse all existing drugs that can reverse the directions of differentially expressed genes from the RNA sequencing data, while also treating the patient's selected phenotypes. From the RNA sequencing data, I was able to obtain a list of all differentially expressed genes between the CHAMP1 samples and the control samples. There are 117 genes that were upregulated and 93 genes downregulated in the CHAMP1 samples compared to with the control samples. The patient actionable phenotypes were listed below. So this 2H query loops through the 210 genes and the 8 phenotypes to find drugs or chemicals that affect both conditions that satisfy both conditions. And the results are 219 chemicals and drugs that meet both conditions. And then I combine, train, categorize, and filter them. After doing a literature review, I curated the chemical list by considering potential side effects and toxicity and safety profile, as well as the relevance of these drugs for this case. So here are some of the consideration for the drug curation. First, I consider them by toxicity level. Are the drugs FDA approved? Are they first-line therapy? Are they chemotherapy? Because chemotherapy may have a lot of trade-off in terms of toxicities. Does this chemical or drug hit the important gene targets? And next, I consider patient phenotypes. Does this drug may treat a particular phenotype for this patient? Does this drug have side effects that potentially aggravate patient symptoms? Then I also consider if there are enough preclinical evidence. And then thinking about should this drug or compound be recommended to the patient directly, or should it be further tested in other assays? After the drug curation, I found that I came up with metformin meet several requirements. So I found that metformin has a great potential to be repurposed for this disorder. A little bit of background about metformin. Metformin is the most common anti-diabetic medication, and I found that this drug can reverse several of the gene expression in the CHAMP1 patient-derived fibroblasts, while it has also been shown to improve cognitive functions in children with intellectual disabilities. Metformin mostly exert its potent hyperglycemic effects by acting on the mitochondria complex 1 activity, and it activate the AMPK and inhibit the mTOR inhibition of the mTOR signaling, eventually decrease hepatic gluconeogenesis and increase insulin sensitivity. Dysregulation in the mTOR signaling has been observed in several neurological disorders, and many studies have elucidated different mechanisms where metformin disrupt this mTOR pathways, and it has neuroprotective and neurogenesis effects, and which raise a strong interest in repurposing this safe and effective anti-diabetic drug for neurodevelopmental as well as for neurodegenerative disorders. In cell culture studies, metformin protected cortical neurons from apoptosis, promote cell renewal of neural precursor cells, supported neuronal differentiation and neurite growth in human stem cells. In animal studies, metformin has been shown to protect mice neonatal brain from injury, enhance spatial memory, and increase lifespans. In humans, metformin use is also strongly associated with slower cognitive declines in older adults, promote brain repair in pediatric brain tumors patients who receive radiation therapy, and improve cognitive functions in individuals with Fragile X syndrome, the most prevalent form of inherited intellectual disability. And this medicines also cross the blood-brain barrier. It also has been demonstrated to have anti-seizure properties. So all these evidence strongly support the rationale to repurpose metformin to improve cognitive functions in this participant. So in conclusion, I have presented a workflow to analyze this patient, analyze CHAMP1 patient RNA sequencing data for drug repurposing with Medikanren. The two-edge query can be used to find drugs given the bunch of genes and phenotypes that we want to influence. Metformin has emerged as a potential drug candidate for drug repurposing for these disorders. About a year ago, we had a plan with Dr. Christine Grimswood at UC Davis to test metformin on their mice model to see if it's improved their developmental delay phenotypes. But we didn't get past the budget stage to fund the study. There are a few things we can improve in this workflow. We may be able to obtain a more relevant cell types or tissues such as neuronal cells or microbrain organoids. We can better design the experiment with more relevant functional tests. In terms of Medikanren, we can improve on having a more robust and automated way to rank and filter the drug results. And that's all I have. And thank you so much for listening. And I wish you have a wonderful time at the conference.